Just over 40 years ago, I started training as a medical laboratory technologist. And during my 5-year apprenticeship, it wasn't actually a university course in those days, I became fascinated with the discipline of blood transfusion medicine and an unusual family of blood group molecules called glycolipids. As seen in this slide, glycolipids have a sugar part, the glycan, and a lipid part. And the lipid part is typically inserted into the cell membrane, anchoring the molecule at the cell surface. But glycolipids, they're no ordinary molecule. They have a fascinating ability to hop into and out of cells. And little did I know at the time, but glycolipids in their later evolution into code technology constructs would become my lifetime research. The first 20-year phase of my research concentrated on trying to understand how natural glycolipids caused unusual blood types and their involvement as receptors for antibodies and microbes. This slide shows a selection of the journal papers in the areas that are in the field I was working in at the time for quite a few years. My research involved a lot of biochemical, serological, genetic, and structural analysis. And this allowed me to discover and unravel a variety of issues, including the genetic and chemical basis of multiple blood types. And ultimately, this research would lead to the discovery of the 31st human blood group system a few years later. From this natural glycolipid background, which still continues today, and the development of a significant global network of research collaborators, the foundations for the second 20-year phase of my research were well established. It was in 1996, after returning to New Zealand from a postdoc in Sweden, that I had the idea that perhaps there was a commercial opportunity for natural glycolipids. I was wrong. After 4 years of investigation, I figured out that natural glycolipids were not viable to make commercial products. So together with my collaborator, Nikolai Bovin, we developed synthetic glycolipids. When the first 5 prototype synthetic glycolipids arrived in my lab, we discovered one instantly dissolved in water, so we put that one aside, but the others behaved like they were expected to as natural glycolipids, and they required drying down onto salts to make them dispersible in a process that we had developed over the preceding 4 years. These latter constructs work just like natural glycolipids, but then we remembered we'd put one aside in the fridge. So we pulled that one out, and I think you can guess it. It was perfect. Overnight, we went from working with complex natural glycolipids, which were onerous to isolate, purify, and handle, to having a virtually unlimited supply of rapidly dissolvable or dispersible synthetic glycolipids. In this slide, on the top, you can see a natural glycolipid, and beneath it, you can see the synthetic glycolipid. You'll note that the synthetic glycolipid beneath has a spacer in between the glycan head group and the lipid tail. And it was this spacer that completely changed our world, and it changed the way the molecules behaved. So we called these new molecules function spacer lipids, the function being the head group, or simply code constructs. The method was quite crazy. We simply disperse code constructs in buffer, add cells, and leave them for an hour. That is it. During the incubation period, the code constructs spontaneously self-assemble into the cell membrane, and the cell is harmlessly labeled with new functions and features, and sometimes even superpowers. Over the next 10 years, the water dispersing spaces of code constructs morphed into more advanced and biologically enhanced versions. And the functional head groups expanded from just glycans to include almost anything. This slide shows some variations of FSL constructs. Using a Lego man representation of the FSL and schematics below, it can be seen that the same functional head represented by the yellow Lego head can be presented by a variety of different spaces, the red body, and lipid tails, the legs. So with just 7 different core spaces and 3 different lipids, A single functional head can be presented in at least 21 different variations. And that's just the beginning bit, and each with a potentially different biological functionality. Today, we have made hundreds of different code constructs for ourselves and our collaborators, and they've been used to attach almost anything biological or non-biological onto living cells, bacteria, viruses, liposomes, and even to modify non-biological surfaces, including plastics, metals, and glass. A couple of examples of code technology as used by us and licensees throughout the world include code molecules have been injected into tumors and are being used as a potential cancer vaccine therapeutic. The licensee BioLine has completed their phase 1/2A human clinical trials, and they are starting phase 2 very soon. The code molecules have also been used in a range of transfusion medicine products, both as quality control systems and diagnostics. And there are several other new products in the pipeline, including COVID-19 antibody diagnostic. Code molecules are regularly being used as research tools and used in a variety of applications, including immobilizing and labeling viruses and cells, through to modification of nanofibers and labeling liposomes, the latter being of current interest for my research team, as well as a range of research immunodiagnostics. With the ability of code to gently label almost any living cell, virus, liposomal surface with almost any small molecule, there are some really interesting new applications for the technology, including novel tools, diagnostics, therapeutic, and commodity use products that are in the pipeline. We often say the potential applications for code technology are limited only by your imagination, and that the best uses for it will be discovered by others, hence why we provide an R&D toolbox to the community. Some interesting publications, in fact, one appeared on my desk just this morning, using code technology construct reagents are constantly emerging in the academic literature, and more appear each year. My career is rather unusual in that I have blended academic and commercially oriented research objectives. I strongly believe there is a place for commercialization of research, which by another name is innovation. And by making product widely available through commercialization, our research is able to have its greatest impact. My mission for my final 20-year phase is to, and I quote the joint Code Biotech and AUT Centre for Code Technology Innovation mission statement, where we plan to ignite a revolution with code surface modification nanotechnology by enabling our partners to enhance existing and create next generation products with the technology to shape life. We are making good progress, and I hope this will make me truly worthy of the honour of being elected a Fellow of the Royal Society of New Zealand. Thank you.